In this section, we'll talk about a group of organisms called the Pseudomonads. It's Pseudomonas and other related genera. We won't talk about all of the genera in here, but we will talk about most of them because they are either very important causes of infection like Pseudomonas and Burkholderia, or they're emerging causes of infections like Stenotrophomonas and Acinetobacter. The first organism we're going to talk about is Pseudomonas aeruginosa. These are gram-negative rods. Remember, you can distinguish them because they are oxidase positive, whereas the Enterobacteriaceae are going to be oxidase negative. You can see the gram-negative rods here visualized by microscopy. They are found in soil, water, and hospital environments and are generally not considered to be part of the microbiome. The genus Pseudomonas encompasses a lot of different bacteria that have a lot of different functions. Some of them are actually beneficial soil microbes that help with plant growth. Some of them can be plant pathogens, there can be animal pathogens, and of course human pathogens. What we are seeing now is that Pseudomonas aeruginosa in particular has really established itself in hospital settings and is a very common cause of hospital-acquired or nosocomial infections. They have very simple growth requirements. I worked with Pseudomonas aeruginosa for the first time as a postdoc. I currently work with it now in my lab. And I came from a spirochete background in my PhD, which the spirochetes are horrible to grow. They take forever. They're very, very slow. They're very, very specific. And what I couldn't figure out was I was working with Pseudomonas and Klebsiella and Staph aureus, and everything that I was trying to grow was contaminated with Pseudomonas aeruginosa. And I couldn't figure out why, because I was keeping everything sterile. I was using the hood. I was using my appropriate sterile technique. And so I tested every reagent that was in the hood. The water was contaminated with Pseudomonas aeruginosa. It was growing in the water. This bacterium will grow anywhere. They are aerobic, and like I mentioned, they are oxidase positive, which again will allow you to differentiate this gram-negative rod from the Enterobacteriaceae. They produce a number of different pigments, including pyocyanin, pyoverdin, and pyorubin. Pyocyanin is the major one that we'll talk about because it serves as a toxin in the host. Aside from toxin production, they have lots of other virulence factors which can make Pseudomonas aeruginosa a pretty nasty infection. Some of those virulence factors include the ability to adhere. So Pseudomonas have flagella, they are highly motile, they have pili, which help with motility and adhesion. They have LPS, which has the endotoxin and also can help with adhesion. And they secrete a polysaccharide component called alginate that helps them form very robust biofilms that, again, will stick to anything, living or non-living surfaces. They secrete lots of toxins and enzymes. A handful of them are listed here. Exotoxin A disrupts protein synthesis, leading to cell death. Pyocyanin, which is the pigment that gives a culture of Pseudomonas aeruginosa a blue-green color. That compound, pyocyanin, produces reactive oxygen species, which cause DNA and protein damage in host cells and in other microbes. Pyoverdin is used as a siderophore. It sequesters iron, so it can steal iron from the host to use it for its own biochemical reactions. Additionally, a number of other enzymes are produced by this bacterium, including elastases, proteases, and phospholipase C, which facilitate tissue destruction. So they can degrade connective tissue and lipid bilayers. They also have a type 3, this is getting nasty, right? They also have a type 3 secretion system. They inject a couple of enzymes called exoenzyme SNT into eukaryotic cells, which will kill those cells. So I mean, I guess if we were to assign a purpose in life to Pseudomonas aeruginosa, it's trying to kill you. And everyone's favorite, actually it's my favorite, antibiotic resistance. That's what my research lab studies the most. And so there is a lot of inherent antibiotic resistance. So just based on the biology of Pseudomonas aeruginosa, it is inherently resistant to chloramphenicol, but it has a lot of acquired resistance as well through plasmids and other mechanisms of genetic exchange with other bacteria. Again, very, very commonly we see Pseudomonas aeruginosa as nosocomial infections or infections in the immune compromised. The most common manifestations of Pseudomonas infection in an otherwise healthy individual would be in patients who have burn and surgical wounds. I had a student, the first semester I taught here, I taught a class called Revenge of the Killer Microbes, one of the upper division GE classes. They do a presentation on a disease, and the student chose Pseudomonas aeruginosa, and they have to produce a pamphlet. So she produced her pamphlet, and it had a picture of a surgical wound that was infected with Pseudomonas aeruginosa. And I sat back in my chair looking at this pamphlet going, I bet that's her wound. That's her surgical wound. She put a picture of her own wound on this pamphlet. And sure enough, it was. And so she had gotten back surgery at one of the hospitals here in Fresno, and every patient operated on by the same doctor that day and the next day all got surgical infections with Pseudomonas aeruginosa. In terms of burn wounds, those are even worse. When a burn wound gets infected, especially if it's a large burn, it can be a death sentence for the patient. I did my postdoc in San Antonio at the U.S. Army Institute for Surgical Research in the Dental and Trauma Research Detachment at Fort Sam Houston, Texas. Fort Sam Houston is home to the San Antonio military medical complex. It's one of the premier burn units in the country. And so not only soldiers who get burn wounds in combat, but also civilians who get burn wounds are admitted to the hospital there. And my postdoc advisor, one of his favorite stories that he liked to tell us when we were new to the lab was that he was called down to autopsy because they wanted to give him the clinical sample to see what the infection was that had killed the patient. The patient died of an infection. It was a patient with what they called TBSA, total body surface area, burns of 35%. So 35% of the patient was burned. And he said he walked in there and he looked at the patient who had passed away, and wherever she had the burn wound, it was green. And he knew, he didn't even have to take the sample. He took the sample anyway, but he didn't have to, but it was a Pseudomonas infection. So lots of studies have shown that if you have a burn infection and a surgical infection, or a burn wound and a surgical wound of a similar size, if you get an infection in the surgical wound, you can tolerate a much higher level of bacteria, and it'll often just form a localized infection that's really hard to get rid of, but doesn't go systemic. In a burn wound, as little as 10 bacteria can be in a burn wound and lead to death of the patient. So burn wounds are really just a type of wound that makes a patient much more susceptible to infection. So if you find yourself working in some sort of emergency medicine, any sort of burn unit, you have to be just so careful to prevent your patient from getting an infection. Other types of infection caused by Pseudomonas aeruginosa can include pulmonary infections. This is fairly common in our patients with cystic fibrosis. And, I've never seen it, but I've heard that in a patient with cystic fibrosis, if they have a Pseudomonas aeruginosa infection, as they're coughing up all of that sputum, you can actually see that it's green because of the piocyanin. It can also cause urinary tract infections, ear infections. And that can be pretty mild. If you've ever had an external ear infection that they sometimes call swimmer's ear, that's going to be most likely a very superficial Pseudomonas infection. However, it can, of course, become invasive, destroy the cranial bones, etc. Eye infections, I think I told you already, I don't like eyes. I think they're disgusting. One of my grad students knew that, and her favorite thing to do in her presentations was to try to put a picture of an infected Pseudomonas eye, and I'm just like, whoa. And it can also, of course, cause bacteremia. It's a gnarly, gnarly pathogen. I really like it. So here, what we see are some different types of wounds. On the left, we see a really deep, like gangrenous wound. On the right, more of a skin irritation. It depends on the host. It depends on the number of bacteria that get in. It depends on a number of different factors, whether or not you're going to end up like the person on the left or the person on the right. This headline I thought was interesting. There was a nasal spray produced by CVS, and it turned out that the nasal spray was contaminated with Pseudomonas aeruginosa. So they had to pull it, obviously, from the market. Remember, on the previous slide, we saw that Pseudomonas can cause pulmonary infections. So basically, if you've got a nasal spray, you're pretty much injecting the bacteria almost exactly where they want to be in order to cause a pulmonary infection. In terms of diagnosis and treatment, they grow on everything. If you grow them in a liquid culture, they will grow near the air-liquid interface, and they'll often they want to form a biofilm so bad. They love forming biofilms. So what you'll often see in a liquid culture of Pseudomonas that's growing in just a standard test tube, you'll see a ring where the liquid culture meets the air, meets the glass, if you can kind of picture right where that is. And there will be like this solid chunk that's left behind when you pour out the liquid culture because it'll form that biofilm. What you'll also sometimes see on the surface of the liquid culture is what looks like a film. Like if you've ever heated milk and it kind of gets a film on it, it's like that. And that's called a pellicle. And it's almost like a little air liquid biofilm kind of. We also have Pseudomonas isolation media. So if you suspect a Pseudomonas infection, you can plate the infectious material on Pseudomonas isolation media and the Pseudomonas will grow. They grow rapidly overnight. You're going to see colonies. They are beta hemolytic. If you grow them on a blood agar plate, they will be able to completely lyse the red blood cells. They form green colonies, especially if you grow them on some of the special Pseudomonas isolation agar. They form these really beautiful blue-green colonies. But even on standard media, they will eventually produce that green colony because of the piocyanin. And they have a grape-like odor. And so sometimes when I tell students this, and I am sorry I can't see your faces, but when I can see your faces and I tell you that, y'all kind of like turn up your nose at me a little bit and you make a face like, ew. Or you make a face like, what does a grape smell like? And so I don't mean that it actually smells like a fresh grape. I mean it smells like that fake grape flavoring, like a grape Jolly Rancher or grape cough syrup. It's just that weird, like fake grape. That's what Pseudomonas smells like. And people don't believe me until they either work in my lab or they go out into a clinical setting. So a student that was enrolled in my class last fall sent me an email, and I won't go into too much detail because I don't want to, spoil it or give it away for her, but she works in a particular type of clinic. A patient came in who was complaining of an infection. When the patient left, she told the doctor, hey, was that patient's infection Pseudomonas aeruginosa? And the doctor said, yeah, how did you know that? He was really surprised. And she said, well, because it smelled like grapes and my med micro professor told me it smells like grapes. And he was like, yep, that's it. So I'm telling you, it smells like grapes. You'll smell the fake Jolly Rancher and you'll know Pseudomonas aeruginosa. Okay, people who grew up in Texas though, because I first started working with this in Texas, they say it smells like tortillas. I don't see it, like corn tortillas. Plus, I love corn tortillas. I'm not letting anybody ruin corn tortillas for me. I don't care about Jolly Ranchers, so that's fine. Because of the inherent and the acquired antibiotic resistance in Pseudomonas aeruginosa, we often want to treat with combination therapy. However, this bacteria is rapidly growing. It's able to acquire resistance really quickly. So what's best is prevention of infection. Multi-drug resistant Pseudomonas aeruginosa is considered to be a serious health threat by the CDC. Nearly 33,000 cases in 2017, over 2,500 deaths, $750 million in healthcare costs. Nasty, nasty pathogen. Closely related to the genus Pseudomonas is the genus Burkholderia. They were originally classified as Pseudomonas, so sometimes when you see old journal articles or old texts, instead of Burkholderia cepaceae or Burkholderia pseudomallii, it'll be Pseudomonas, but molecular and biochemical analysis has shown that they are actually two different genera. Burkholderia cepaceae causes complex opportunistic infections, again, particularly in our cystic fibrosis patients. So I'm not sure how familiar you are with cystic fibrosis. It's caused by a mutation to a single gene that's involved in chloride transport, and so your patients end up getting a buildup of really thick, sticky mucus in their lungs, so it can cause a patient to suffocate. The only way to cure a patient of cystic fibrosis is to do a lung transplant. But because their lungs are so full of this sticky, gross mucus, they are super, super prone to respiratory infections. And so things like Staph aureus cause a lot of cystic fibrosis infections, Pseudomonas aeruginosa, but also this Burkholderia cepaceae. And the problem with this one is it's so contagious and it's so difficult to get rid of that a patient who has been approved for a lung transplant but pops positive for this Burkholderia infection will no longer be able to get a lung transplant. So it's a contraindication. Any patient who has a Burkholderia infection cannot get a lung transplant. And if they can't get a lung transplant, they could die. The average lifespan of a patient with cystic fibrosis is into the early 20s. So to extend the life, you need the lung transplant. You need to not have a Burkholderia infection. There was some like teeny bopper movie where like they fell in love, but they couldn't get close to each other because they had cystic fibrosis. It's because they have these infections. I don't know what the movie is. I'm not going to look it up, but you can if you want. Burkholderia pseudomallii causes an infection called melioidosis. This is a cutaneous infection that can sometimes go septic. It's not very common. It can also cause pulmonary disease that can be mild, or it can progress all the way to necrotizing pneumonia, where basically the lungs are being destroyed by the bacterium. Now, one way that you could pretty easily differentiate between Burkholderia and Pseudomonas is that Burkholderia is susceptible to trimethoprim sulfamethoxyl. Sometimes this is abbreviated as TMP SMX or SMZ. It really depends on who's doing the abbreviation. The generic name would be something like Bactrim. So Burkholderia is susceptible to TMP SMX or SMZ, whereas Pseudomonas aeruginosa is resistant, because Pseudomonas aeruginosa is pure evil. Stenotrophomonas, again, not one that's super common, but there's a really great documentary that I watched. I saw it on Netflix, but last time I checked, it was available on PBS, and it was free through PBS. It was produced in 2013, so it's a little bit old, but I'm a big nerd, and I like this kind of stuff, and it was called Hunting the Nightmare Bacteria. And I had never heard of this genus before, but this genus was mentioned in that documentary, so I highly recommend watching that. But anyway, Stenotrophomonas here in a patient who is neutropenic, meaning they have decreased levels of neutrophils. So this is an 8-year-old Chinese girl with acute myeloid leukemia. Leukemia is the pits, absolutely the worst. Awful, awful disease, totally suppresses the immune system. So of course, this poor patient has a complex history of recurrent fungal and bacterial infections, including pulmonary aspergillosis. We'll talk about aspergillus when we talk about fungi, but septicemia with Klebsiella, Enterobacter, Staphylococcus, Streptococcus, and Bacillus. She was receiving combination therapy with meropenem and amikacin, so a carbapenem or beta-lactam and an aminoglycoside, which inhibits protein synthesis. During a period of severe neutropenia, so decreased neutrophils, she became bacteremic with Stenotrophomonas multifida, which was sensitive to TMP-SMX. Over the next few days, she developed painful red skin lesions. This bacterium was isolated from one of the lesions. Treatment with TMP-SMX led to a reduction in the lesions. So this case is trying to illustrate the fact that this genus can cause infection in immune-compromised patients who are receiving a carbapenem antibiotic. So one of the dangers is, I told you, carbapenems are one of those last-line beta-lactam antibiotics. Well, Stenotrophomonas is one of the few gram-negative bacteria inherently resistant to carbapenems. It is also inherently resistant to aminoglycosides, but it is susceptible to TMP-SMX. So when you have a patient who you have given combination therapy, 2 different mechanisms of action, and you still are popping positive with an infection, perhaps Stenotrophomonas. Acinetobacter is another genus belonging to the group of Pseudomonads. It is considered to be a saprophyte, so it's found in the soil, and it can be found in moist or dry soils. The species that causes most human infections is Acinetobacter baumannii. This is not a new infection. It's more of an emerging infectious disease. So there were always a handful of patients every year who had infections with Acinetobacter baumannii. But it wasn't until 2002 when soldiers started coming back first from Afghanistan and then from Iraq in like 2003-04 that we started to see really high levels of this Acinetobacter baumannii. So it causes opportunistic infections of wounds, urinary, and respiratory tracts. And because we started to see it so commonly in the soldiers, it got the nickname Iraqibacter. Because this is a soil-borne organism, the reason that we started to see more and more infections in our soldiers is that part of the combat strategy of the Iraqi or Afghani fighters was to bury IEDs, improvised explosive devices. When the IED goes off, it blows soil up. And so a soldier would be wounded by the shrapnel or by the concussive force and have soil driven into the wounds. The goal of battlefield medicine is to prevent the soldier from dying on the battlefield. And in fact, battlefield medicine has gotten so good that something like 90% of wounded soldiers survive their wounds. It has to be a really significant wound directly to the brain or the heart, or to cause just too much damage in order for the patient to not survive the wound. But really what we're looking at is stop the bleeding, not keep the wound clean. And so these wounds would not be cleaned. They would just be closed up to basically prevent the soldier from dying. The soldier would then be sent from wherever they were injured to the major operating base in the country. Then they were generally evacuated to Ramstein in Germany. Then they were sent to Walter Reed in the US, or sometimes they were sent to San Antonio, Fort Sam Houston. And this can take 7 to 10 days. So basically what you have is a soldier that you've kept alive who may or may not be beginning to heal, but you have this bacterium, this Acinetobacter, inside of the wound that is continuing to grow and can cause a very significant infection. So this came out in 2005. Soldiers are coming back with multi-drug resistant Acinetobacter baumannii. So infections, multi-drug resistant infections were first noticed on a U.S. Naval hospital ship and then at Walter Reed. Since then, more than 250 cases have been identified, including 18 documented cases of nosocomial transmission. So basically what you're looking at is along this whole route of soldiers being evacuated from Iraq or Afghanistan, they are often being, pumped full of antibiotics. They're being exposed to other organisms because they're in hospital transport, they're in hospitals, they're on a base. And so it's this perfect breeding ground to now confer antibiotic resistance to this bacterium. So now it's a major cause of antibiotic-resistant infections, especially in hospital settings. So the CDC considers carbapenem-resistant Acinetobacter to be an urgent threat. So we went from 250 cases identified in 2005 to 8,500 cases leading to hospitalization. 8,500 cases in hospitalized patients in 2017. That's a huge increase. 700 deaths, $281 million in healthcare costs. This is a really significant emerging issue. So military superbug, quiet civilian epidemic, Acinetobacter infections associated with the war in Iraq, unusual cases. So again, it's not a new organism, it's not a brand new infection, but because of medicine and because of human activity, it's becoming more and more prevalent.